So Kaycee, welcome.
Thank you.
To get started, why don't you tell us a little bit about your background?
I did medical school, residency, and my fellowship all in California at UCSF. My residency was internal medicine, and then I followed that with a 3-year geriatrics fellowship, which also combined a master's in clinical research. I went to Wake Forest University in North Carolina, in part because I had the opportunity there to start a memory disorders clinic. So I spent 13 years at Wake Forest. I led a lot of clinical trials as either a PI or worked on them as a co-I, and was the principal investigator for our clinical core for the NIH-funded Alzheimer's Disease Center. And after about 13 years, I really felt pulled in so many directions, and I thought, wouldn't it be great if I could go somewhere where we're all playing on the same football team, and everybody's goal on the team is to get the ball from here to the goalpost down there. And that was one of the things that really drew me to Pharma and to Genentech and Roche specifically.
So your role at Genentech, so there for 7.5 years, where did you end up in terms of, you obviously rose through the ranks over that time. What was your role at the end of that period?
So I was a global development leader, which basically is a team lead on a usually phase 3 program and whatever studies are in it. The global development leader essentially leads the cross-functional team that puts the strategy in place for how we're going to get this molecule from, through the clinical trials and up to FDA approval or other health authority approval and into the market.
What are some of the real challenges that pharma companies and pharma execs face, particularly in respect of Alzheimer's disease trials?
I think in global development, some of the challenges are simply the fact that it's global, right? And you have different health authority requirements in different regions and different rules about data, confidentiality, and how that data can be moved and transferred. The stakes are high, and so you have to really be forward-looking and think about not just what you're doing now, but how what you're doing now is going to set you up for what you're trying to achieve in the future, and making sure you're prepared when you get there. There's a lot of things that I don't think I fully understood when I was in academia. You move into pharma, which is highly regulated, and there's a lot of rules, but for a lot of good reasons.
And true to say that once a compound gets to you, the chemistry part's all sorted, and so interested in your thoughts around the comment that a lot of the problems are around the endpoint data quality.
So the trial design is important, but the execution is critical, because if you don't collect the right data or it's not of the right quality, you might have a drug that works, but either you couldn't show that it worked, or it worked, but maybe not to the standard or the quality. There's quality issues that call it under question. So I think it's absolutely important to make sure that you not only have the right endpoints, but you're collecting them in the right way, and that you achieve good quality for especially the primary endpoints, of course.
And so in that context, in that data quality context, interested in your thoughts about the role of technology as the industry is moving forward. So how you've seen that the role of technology change, I suppose, during your 7 years at Genentech, but also how do you envisage that'll change into the future?
Technology is advancing so quickly, and changes are being made. When I first started doing these trials, it was all still pencil and paper. Going forward, I would love to see 2 things happen. One would be more sensitive tests that aren't just paper copies, digital versions of paper copies, and those are tried and true, but there's a lot of noise in some of them, or they weren't designed to capture the stage of dementia that we're now most interested in. They work great for mild or moderate dementia, but not so much for detecting preclinical AD. So I think we need more precise tools that can capture subtle changes over time and then be validated against important clinical endpoints, obviously. And then the second thing is that our trials are massive, and so we have to find more efficient ways to monitor the quality of the data. And I think this is one place where capitalizing on the advancements in AI might be helpful. An AI algorithm could identify which assessments need to be reviewed by a human and quality checked.
And just to go back to something you said, when you talk about the stage of dementia and the the changing needs in terms of the assessments. You come with a unique view from within pharma of where the focus is, and the types of patients we're trialling with new treatments now are getting earlier and earlier, aren't they?
They are. There's increasing understanding that the earlier we can intervene with disease-modifying drugs, the better off the prognosis and overall life course will be for those patients. So we've got to figure out how to measure cognition at the very earliest changes so we can identify the right patients for these trials. And that's super tricky because you can't screen everybody in a clinical trial and bring them in for a several-hour screening visit. So I think pre-screening is going to become hugely important, and being able to do that efficiently and at the community level. I also don't want to forget all the people who are currently suffering with dementia, and my heart goes out to families who they're looking for clinical trials and their loved one already has moderate stage dementia, and there's often nothing for them because they've missed the window. We still need to be able to capture the whole range of cognition, but in terms of really making an impact downstream, I think moving upstream is going to be critical. Yeah.
One of the problems is, as you mentioned, is really finding those patients, right? Because a lot of these people won't necessarily recognise that there's changes going on inside their brain. And so when you're designing a trial in your previous role, how much time do you spend thinking about how are we going to recruit these patients?
A lot. I think it depends on which patient. So if you're thinking about prodromal to mild, recruitment's important, but it's not that hard. I think the field has figured out how to do it. The biggest focus there would be, how do we increase diversity and making sure that we have representative patient populations and getting to the patients that we don't usually present for clinical trial. Then the other part is, well, how about the people who don't yet know they have a problem? And nobody can see that they have a problem. And if we really want to do prevention, how does that look? And I think trial design has to change. The whole paradigm for how we do this. I mean, we can't find people and follow them for 10 years to a clinical endpoint. So I think blood-based biomarkers are going to become critical, and they are becoming critical. Ideally, we could pair a blood-based biomarker with a very brief cognitive test. And I think what the goal would be is to match the approach with the desired endpoint. There's no one-size-fits-all thing, and so it really depends on what are you trying to achieve, what are your goals. And I think for different companies, if cost minimization is a priority and the budgets are lean, then you pick a different approach. In other scenarios where the cost of screening maybe isn't the primary driver and speed is a bigger driver, then you might choose a different paradigm. So I think it all depends on what the goal is and what you're trying to achieve.
But such an opportunity to, if we can use technology to reduce the cost of that screening in the community, such an opportunity to have different ways of finding patients and then directing them into trials.
Yeah, and I think I see a twofer here. So in addition to making screening more efficient with technology, we can also reach those pockets of people, the more diverse communities that I said we didn't normally get. So if we could utilize technology to bring a more hybrid approach and be able to reach more people who wouldn't normally be eligible for clinical trials because they're 3 hours away and they can't come that often to site visits, then we could open up the doors to getting access to more people.
So now that we've got 2 drugs approved, what your expectation in terms of what's the amount of investment by pharma companies in Alzheimer's disease, do you expect that to increase?
Absolutely. This is one of those situations where now the proof of concept has been achieved. We can make a dent, we can make an impact, there is something that's going to work. We should see a lot of investment in different MOAs, in combinations, in next generations of the mAbs. So I'm really excited about all the things that are going to be bubbling up the pipeline.
Because it's not just amyloid, is it?
No. I mean, I think now that we can tackle amyloid, it's prime time to then see what can we do with tau. We got to do something. But there's also lots of other targets that were less important maybe than amyloid initially. But now that we've figured out what we can do about amyloid, I think it's prime time to start turning our attention to some of these other things, because we can only make so much progress with amyloid. What if that's 30 or 40 or 50% effect, there may be a ceiling there.
And then just more generally looking forward, what are you most excited about in terms of Alzheimer's disease research?
I think maybe there's been a pessimism for so long. It's been over 20 years since we've had any new therapies. And now it's almost like there's this breath of fresh air. Wow, like a lot of excitement behind AD and the fact that maybe we're finally making some progress. And I think doubling down on pathophysiology and other targets. So I'm really excited for the future of patient care and what we can do. I think we're just at the very beginning. It feels like in AD we are where cancer or hypertension or cardiovascular disease was about 20 years ago. Like we're just on the precipice of, I think, an explosion of understanding. That understanding and those, new drugs then open up a lot of avenues for various different, sequential treatments, combination treatments, things that'll actually improve patients' lives. And that's what's so exciting about this, because I feel like when I talk to patients now, I can say this might actually slow your disease, as opposed to being the symptomatic drugs, which might make you feel better while you take it, but isn't changing what's actually happening in your brain.
Your experience in terms of being able to go from as a prescriber, as an academic researcher, as a pharma executive, and to bring all that to bear in terms of what we're trying to achieve in Cogstate, I think we're really lucky to have you.
Thank you. I'm happy to be here.